Thank you everyone for joining us, and welcome to the Cancer Research Institute's webinar series, Breakthroughs in Immunotherapy. Today's date is Tuesday, September 30th, 2014. Today's webinar is titled Immunotherapy, the Future of Prostate Cancer is Here. Today's webinar is made possible through the generous support of Dendreon Corporation. Dendreon is a biotechnology company whose mission is to target cancer and transform lives through the discovery, development, commercialization, and manufacturing of novel therapeutics. The company applies its expertise in antigen identification, engineering, and cell processing to produce active cellular immunotherapy, or ACI. ACI product candidates are designed to stimulate an immune response in a variety of tumor types. Dendreon's first product, Provenge, or Sipuleucel-T, was approved by the U.S. Food and Drug Administration in April 2010. Dendreon is exploring the application of additional ACI product candidates and small molecules for the potential treatment of a variety of cancers. The company is headquartered in Seattle, Washington, and is traded on the NASDAQ Global Market under the symbol DNDN. For more information about the company and its programs, visit www.dendreon.com. Thank you, Dendreon, for your support. My name is Brian Brewer. I'm Director of Marketing and Communications at the Cancer Research Institute. Cancer Research Institute is the world's only organization dedicated exclusively to harnessing the immune system's power to conquer all types of cancer. We fund scientists around the world whose work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 45 minutes, you'll have an opportunity to hear firsthand from an immunotherapy expert how the treatment of prostate cancer today is undergoing a revolution thanks to these breakthroughs. After a 25-minute presentation from our speaker, we will open the discussion to questions submitted by you. You can pose your question at any time throughout the webinar by typing in the Q&A box on your screen. Today's webinar is being recorded and will be made available for viewing on the Cancer Research Institute website and on our YouTube channel. Now it's my pleasure to welcome today's speaker. Dr. Charles Drake is an Associate Professor of Oncology, Urology, and Immunology at the Johns Hopkins School of Medicine in Baltimore. He also serves as co-director of the Prostate Cancer Multidisciplinary Clinic. His areas of expertise include immunotherapy and the diagnosis and treatment of cancers of the prostate, kidney, bladder, and testes. His work is widely published. He is the winner of numerous awards for his research and his scholarship, and he holds several patents. We're honored that he could join us here today. Dr. Drake, welcome.
Thank you. So we'll get started. Today's webinar will focus on the basic principles of immunotherapy for prostate cancer, and we'll discuss several interesting technologies that are either FDA approved or in clinical trials development. These are my financial relationships as is required. You can read them at your leisure. We'll talk about several agents that are not FDA approved for the treatment of prostate cancer at this time, specifically ProstVac VF, which is a vaccine, and ipilimumab anti-CTLA. So today, in today's talk, we'd like to first talk a little bit about immunotherapy or an immune response in a more broad setting. Talk about how it works. We won't spend too much time on that, but enough to have a background so that you can understand the rest of the presentation. Then we'll focus on two cancer vaccines, one which is FDA approved and one which is currently undergoing phase 3 evaluations. These are Sipuleucel-T and ProstVac VF. Next, I'll talk about a large randomized phase 3 trial involving the agent ipilimumab, which is an antibody to CTLA-4 and which is an immune checkpoint blocker. Works a different way than the cancer vaccines that we talked about earlier. I'll talk a little bit at the very end about the idea of combining immunotherapy with hormonal therapy, and then show you a sort of challenging slide talking about how we think about involving immunotherapy in the treatment paradigm for prostate cancer moving forward. So when we talk about an immune response to cancer, most of the time we're thinking about an adaptive immune response. By an adaptive immune response, this is an immune response that involves lymphocytes, T cells, and B cells, and which eventually, if we're lucky, can give rise to memory. And the key take-home point to remember here is that the cell that starts an immune response is called a dendritic cell, or an antigen-presenting cell. These cells normally live in your body in a resting state, kind of constantly sampling their environment. They do this by a process of pinocytosis, kind of drinking up the environment around them. And normally that's all they do. However, when they're activated either by a bacterial product or on purpose, say by a vaccine, what they do is they mature into these cells that we call antigen-presenting cells. They grow these rather beautiful projections we call dendrites. This is why they're also called dendritic cells. And now their job is to be the cell that starts an immune response. They do this by activating two sets of lymphocytes, both CD4 cells here, which are helper cells. These are cells that provide help to other cells in the form of cytokines or other chemical messengers. They're actually absolutely required to make antibodies. And these are the cells that are missing in patients with HIV. So CD4s are the helpers. The dendritic cell also presents its antigen to and activates CD8 cells. These are the cells that we really are hoping to get activated in an anti-cancer immune response. And these are the cells that can traffic widely through the body. So again, in terms of a take-home message, the cell that starts the immune response is in fact this guy, the antigen-presenting cell, also known as the dendritic cell. If I use both interchangeably, please forgive me. The cell that does the work, that actually performs the lysis, is the CD8 T cell. These cells are rather amazing. They can traffic widely throughout the body. They can lyse chemotherapy-resistant tumor cells. And they can kill multiple targets in sequence, actually. And again, if we can engender an appropriate CD8 T cell response, the hope is that we can have a meaningful response against cancer. So how can you do that? Well, it turns out that in patients with cancer, the key cell, in fact, the antigen-presenting cells, are often dysfunctional. Sometimes there's not enough of them. Other times they simply are refractory to stimulation. They present the antigen in kind of a negative way. Instead of turning on the CD4 and CD8 cells, in fact, actually turns them off. So if you believe that, one approach to immunotherapy might be the idea of generating these key cells outside the body, outside the immunosuppressive microenvironment in a cancer patient. And to do this, what's done is lymphocytes are harvested, actually leukocytes are harvested from a patient's peripheral blood through a process known as leukapheresis. And in order to turn these cells into a vaccine against cancer, they're incubated with fusion protein that has this substance called GM-CSF, which activates the dendritic cells. It actually matures them into more functional antigen-presenting cells. And then also, to make a vaccine, you need a target. You need an antigen. And in this approach, the target antigen is a protein called prostatic acid phosphatase. So what happens is patients undergo leukapheresis. The cells are shipped to a processing facility where they're incubated with this fusion protein for about 36 to 44 hours. And then they're sent back to the patient for IV reinfusion. And this is the concept of Sipuleucel-T or Provenge, which has been tested and is FDA approved in prostate cancer. It turns out that in any vaccine, you don't need just one dose. You need a prime and then a boost. And in the Sipuleucel-T approach, there are 3 doses altogether given at 2-week intervals. So the entire process takes around 6 weeks or so in total. Evidence that this works was derived from a clinical trial. This is the pivotal trial. It was called IMPACT. In this trial, about 500 patients were randomized 2-to-1 to either receive the active vaccine or a placebo. Patients were tested at progression. It turns out that the trial did include a crossover, so patients who progressed were allowed to cross over to the active treatment side. Primary endpoint was overall survival, and as we're all aware, the trial did meet its primary endpoint. This is the survival curves from IMPACT presented in the New England Journal of Medicine by Dr. Phil Kantoff. And you can see in the yellowish lines, this is the survival of patients treated with Sipuleucel-T versus placebo. In the grayish lines, a median survival benefit is in the range of 4 months. It's important to remember who these patients were. This trial involved men with metastatic, castrate-resistant prostate cancer that was either asymptomatic or minimally symptomatic. In addition, these men did not have visceral metastases. It turns out that visceral metastases portend a poorer outcome, and those patients might be harder to treat. So this drug is FDA approved, and it's approved for the population was tested in the phase 3 trial. You know, I often get this question, actually, and I maybe even could head it off here, is there any evidence that this drug induces the kind of adaptive T-cell response that we were talking about in the beginning, the kind of adaptive T-cell response that we think could potentially lead to an anti-tumor response? And These data were published in the Journal of Cancer Immunology and Immunotherapy, CII is what we call it, by Nadeem Sheikh. And these data show the production of an adaptive immune response measured three different ways. So on the far left side is the production of antibodies. And the C, or the red bars, are the control group, and the S is the Sipuleucel-T group. And the top set is antibodies to the fusion protein that I just showed you. And the bottom is responses to the native antigen, the prostatic acid phosphatase, which we think is the target in patients of this kind of a vaccine approach. And what you can see is there's pretty good evidence that patients who are treated with this make antibodies against prostatic acid phosphatase. If you take the T cells out of the patients and you mix them with either the fusion protein or with PAP, you can ask if they've been primed, and you can ask that by seeing if they divide or proliferate. And sure enough, you can see that patients who were vaccinated, their T cells, in fact, do proliferate in response to either the actual antigen or to the fusion protein. And not only do they proliferate, they actually make effector cytokines, the kind of cytokines that are important in killing, like interferon gamma. So the bottom line is that there are pretty good data supporting the notion that this vaccine induces an adaptive immune response. And that's something that not everyone is aware of at this time. So what are the takeaways about the first FDA-approved vaccine? It's actually the first FDA-approved vaccine for the treatment of any solid tumor. It's called Sipuleucel-T, so it's FDA-approved. It's indicated for men with asymptomatic or minimally symptomatic castrate-resistant disease. I just showed you a couple seconds ago the data that it induces both antibody and T cell responses. In terms of clinical treatment, it's a well-tolerated regimen. Common side effects include fever, chill, flu-like syndrome that are consistent with the idea that the immune system is being activated, which is good. I showed you the data that there's a survival benefit in phase 3 trials. I just showed you one, but there's several other trials that support this. The downside of using Sipuleucel-T in patients is that there are few objective responses. Very few patients, less than 5%, have tumor shrinkage or a drop in their PSA. So it's challenges as clinicians to explain to patients what the tangible benefit for them is actually. Nevertheless, this is FDA approved. And, we'll talk a little bit more about this at the end, but when we use this in the clinic now, it tends to be one of the agents that we use earlier on in the treatment paradigm, especially before chemotherapy. Although in the pivotal trial, patients could have had chemotherapy. But in general, this is something that we like to use earlier in the treatment paradigm for prostate cancer. So are there other ways to make a vaccine? Yes, there are many, many ways to make a vaccine. I'm not going to tell you about all of them, but I'm going to just focus on a vaccine that's made it to a phase 3 trial, pivotal phase 3 trial that's ongoing now for prostate cancer. And this is a vaccine called ProstVac VF. This vaccine is very different, actually. This vaccine is based on the vaccine that cured the world of smallpox. It's called vaccinia. And to turn vaccinia into a vaccine for prostate cancer, two things were done. One thing was it had to be transfected to express a target antigen, and in this case, the target antigen is different than Sipuleucel-T. The target antigen is in fact PSA, prostate-specific antigen. The other thing is that this kind of a vaccine is by itself reasonably potent, but there are things that you can do to the vaccine to make it more potent. Specifically, a triad, 3 different co-stimulatory molecules were added in order to make the vaccine do a better job at both activating those dendritic cells we talked about, those antigen-presenting cells, and also helping them to attract T cells. So those are those 3 molecules here. The vaccine is injected subcutaneously, and in the end, frankly, immunologically, it does pretty much the same thing that Sipuleucel-T does from an immunologic sense. That is, it activates antigen-presenting cells. They now present the target antigen. It's a different target antigen, PSA, to both CD4 and CD8 T cells. And again, the concept is once activated, these cells traffic widely throughout the body and hopefully slow down tumor progression. Whenever you see a scheme like this, I know what you're thinking. I'm thinking the same thing. Is there any evidence that this might have some benefit or some efficacy in cancer patients? And in fact, there are some evidence from data from a phase 2 trial, again published by Dr. Phil Kantoff in the journal, this time in the Journal of Clinical Oncology. This was an interesting trial. This was a randomized phase 2 trial where men with asymptomatic or minimally symptomatic castrate-resistant prostate cancer that was metastatic were randomized either to ProstVac or to placebo. The ProstVac is shown here in orangish color, kind of yellow color, and the placebo in this is bluish color. And this was an interesting trial. So this trial was not designed to look at overall survival. This trial was designed to look at progression-free survival. That was its primary endpoint. It did not meet that primary endpoint. However, long-term follow-up, largely mediated by a gentleman named Wayne Godfrey, who followed these patients for a long time, was conducted, and that analysis, which was not, again, it's not the primary endpoint, suggested a fairly robust survival benefit in the range of about 7 or 8 months or so. That's not level 1 or even level 2 evidence because that wasn't the primary endpoint of the study. Nevertheless, the data were suggestive. They looked fairly impressive, and so based on that, a large, and I mean large this time, a really large, randomized phase 3 trial was started. This trial has the name ProstVac, and this is a 1,200 patient trial. In this trial, men, again, always focusing on the same group, men with asymptomatic or minimally symptomatic castrate-resistant prostate cancer who haven't had chemo, this is a pre-chemotherapy trial, are being randomized to 3 different treatment regimens. One is placebo. This is the control arm. And in this trial, unlike the Sipuleucel-T trial we talked about earlier, no crossover is permitted. This should lead to a cleaner endpoint of overall survival. Patients in another arm are treated with the combination of ProstVac VF. This is a prime and many boosts of ProstVac over time, along with GM-CSF. That's the same substance we just talked about. This is a cytokine that helps to activate the dendritic cells and do a better job at presenting antigen. One thing that's interesting, in this is sort of an immunological fine point, is we're not really sure exactly how much, this is a pretty potent vaccine, right? So we're not sure exactly how much the GM-CSF was needed. It's a little bit cumbersome. It has its own set of side effects. It's in separate injection intradermally. So one of the questions that I think perhaps the regulatory agencies proposed to the folks running this trial was, do you really need it? Do you really need that GM-CSF? So another arm has the ProstVac VF alone. I should point out that in trials of melanoma, in some trials anyhow, it looked like the subcutaneous GM-CSF actually made the vaccine maybe work a little bit worse. These are data published by a friend and colleague of mine, Craig Slingla. So this is a very reasonable design. It's a large trial, 1,200 patients total, 400 per arm. Enrollment is currently accruing, but these men now, men with metastatic, castrate-resistant, asymptomatic prostate cancer, they live fairly long. Estimated survival based on other recent experiences with these kinds of patients would suggest that they're probably going to live in the range of 3 years or so. So this is not a trial that's going to read out next week because basically the current time it's still accruing. But it is something that we're looking forward to going forward. So what are the take-homes about ProstVac VF? So if it's approved, which will be again in several years, it will be in the pre-chemotherapy setting. It's given intradermally, which is fairly straightforward. There are, just like Sipuleucel-T, vaccines have few adverse events. And just like Sipuleucel-T, in the phase 2 data that I showed you, there were very few objective responses. Although they're not unheard of, they're just rare. This is what's going to happen with ProstVac VF. And again, this is something that we'll be looking forward towards over the next couple of years. Now, in other cancers, we focused a little bit more strongly on another way to activate an anti-tumor immune response. And this involves a series of molecules called immune checkpoints. These are molecules that are on T cells, and they serve to turn the T cell off or to restrain its function. It probably evolved to protect your body from excessive inflammation during an infection. And I think what happened is that tumors hijack them and they use these kind of molecules to escape the immune system. And the way these molecules work, as we mentioned in the very beginning, T cells are very powerful cells. They can kill multiple cells, including your own, which is autoimmunity. And so it turns out that in order to turn one on, a system evolved that makes it that you need two signals to fully activate a T cell. I like to think of this as when we launch nuclear missiles, which hopefully we never will, but it turns out there's not just one guy who pulls the lever, there needs two guys to pull the lever to actually launch a T cell. Those two levers include one that's specific, that's through the T cell receptor. This is a molecule on the T cell surface that's exquisitely specific for maybe one target antigen. So in prostate cancer, we're hoping that it's a T cell receptor that's specific for something like prostatic acid phosphatase, or PSA. The other molecule, the second lever to launch the T cell, is known as CD28, and that sees its target molecule called B7-1, which is on those antigen-presenting cells. So again, for a T cell to launch, it needs both a specific signal, signal 1, and then a non-specific signal coming from that activated APC called signal 2. In the context of cancers, and this happens in many cancers, not just prostate cancer, in fact, it happens very strongly in melanoma, what happens is the T cells upregulate a molecule, a checkpoint molecule, called CTLA-4. CTLA-4 binds to that signal 2 molecule with higher affinity than the normal ligand, CD28. So essentially, the CTLA-4 quite evilly or beautifully hijacks signal 2 and quite simply turns the T cell off. And when I mean off, I mean that it doesn't proliferate and it doesn't exert effector function. And it actually doesn't traffic very well either. So basically, CTLA-4 and molecules like it, like PD-1 and LAG-3 and TIM-3, other kinds of molecules like this, turn off T cells so they can't fully be activated. When I talk to medical students about this, I always say, think about this. Is there anything that you can do about this? And it turns out there, in fact, is you can create through technology, hybridoma technology, very potent and specific proteins called monoclonal antibodies. These bind to their targets with really high affinity, nanomolar affinity, and they can block interaction. So there's an antibody, actually there's a series of antibodies that have been generated against CTLA-4. That little alpha there means anti, so it's like anti-CTLA-4. The ones that are in clinical development are called ipilimumab and tremelimumab. And what happens when patients are treated with these antibodies IV is you block that negative signal, and now signal 2 can proceed forward like that, and the T cell can then go ahead and be fully activated. Always there's a catch, right? So it turns out that these molecules, as I mentioned, did not evolve to protect your tumors from your immune system. What they evolved for was to protect you from autoimmunity or excessive immune attack. So you could imagine that if you give anti-CTLA-4, you might wind up with inflammation, things that end with itis in areas of the body that maybe you don't want them to happen. And the most common include inflammation in the skin, dermatitis, inflammation in the gut, colitis, inflammation in the liver, hepatitis, and occasionally even in the brain in the hypophysis known as hypophysitis. So those are expected side effects. My good friend Dr. Sue Slovin did a great job compiling data from a series of phase 1 and 2 trials of anti-CTLA-4, CTLA-4 blockade in late-stage metastatic castrate-resistant prostate cancer. And these trials involved a number of different doses of anti-CTLA-4, 3 mg per kg, 5 mg per kg, or 10 mg per kg. And in these trials, the agent was either given without or with radiation therapy. And so these are a series of trials. And what Dr. Slovin was able to figure out was that there was some evidence for a PSA response. Anti-CTLA-4 did look like it has activity in metastatic prostate cancer, with the response rate in this data set in the range of maybe 10 to 20% or so. This data set is small, so it wasn't obvious that radiation was adding or subtracting here. But in other experiments, particularly in animal models, it looks like radiation therapy can augment an anti-tumor immune response. So based on those data and also on data from melanoma showing clear survival benefit for this agent, a phase 3 trial was launched. This is known as the 043 trial. We recently published the results in Lancet Oncology. So this trial was different than the ones I just showed you. This is a very late stage trial. So this is for men with metastatic castrate-resistant prostate cancer who have failed or progressed on or after chemotherapy with docetaxel. So this is a late-stage population. Because these were late-stage men, the decision was made to include even men with visceral metastases, though we know that that's a patient group with a poorer prognosis. Men were randomized 1-to-1 to receive either ipilimumab at 10 mg per kg or placebo, preceded by a little dose of radiation to liberate some antigen and maybe start an anti-tumor immune response. As you might have heard, this trial did not meet its primary endpoint. It was a negative trial. So the Sipuleucel-T arm here, so the ipilimumab arm is shown here in the blue, and the placebo arm in the yellow and the orange color. And it was close, actually. The survival difference was about a month, but it was not statistically significant. The p-value that was close was 0.053. And when you see these kind of data, what you often should think about doing is going back and seeing, why was this not a positive trial? And that was done through a number of analysis, but one of the best ways is to just look at the subgroups that were treated and to see if there was any subgroup in which the checkpoint blockade seemed to have particularly good or particularly bad effect. And this is what's called a forest plot. When the groups are to the left of this unity line, it's suggesting benefit, and when things are to the right, it suggests that there's not a benefit. So you can actually look, did younger men or older men have a benefit? It really didn't matter. Did their performance status matter? Yeah, not really so much. One thing jumps right at you at this picture is the presence of visceral metastases. So it's pretty clear that patients with visceral metastases really didn't look like they benefited from this treatment at all. And if you go back and redraw those survival curves by the presence of visceral metastases, it's actually fairly dramatic, right? So in the red and the green are the patients with visceral mets, either with or without ipilimumab. And it's clear these curves are nearly superimposable. So this agent in this setting had absolutely no benefit in this retrospective analysis. If you had looked only at men without visceral mets, it looks like there might have been a better chance of the trial meeting its primary endpoint. The reason I'm telling you all that, so this drug is not FDA approved for prostate cancer, but around the same time, another trial was launched. This is a pre-chemotherapy trial. It's called the 095 trial. This is took men with metastatic castration-resistant cancer, but before chemo, and randomized them either to ipilimumab or placebo. The big difference is this trial excluded men with visceral mets. This is fully accrued, and it should read out probably within the next year or so, actually. So the bottom line is ipilimumab does have a second chance in metastatic castration-resistant prostate cancer. Again, in an earlier group of men, and without visceral metastases, and we're all waiting for those data. So again, takeaways from ipilimumab in prostate cancer: it's not FDA approved. I say it a lot of times. If it does get eventually approved, or really label extended, because it is already approved for melanoma, it would be in the pre-chemotherapy setting. As I alluded, the rate of adverse events is in the range of about 15 to 20, maybe even 25%. These can be challenging to manage, and a series of algorithms have been developed to help clinicians manage these. They generally involve recognizing immune-related adverse events and then treating early on, if appropriate, with corticosteroids. The response rate in the pre-chemotherapy setting, we don't know. It might be significant. And what's really interesting in melanoma, it looks like maybe about 20% of the patients who are treated with this agent are alive at 5 years. Kind of that's what we're hoping for in prostate cancer as well. This is not known yet for either trial, for the 043 trial or for this trial. And it's certainly something that we're looking forward to figuring out going forward. Now, is there anything that we can do, particularly in the early setting, to make the immune system do a better job at recognizing prostate cancer? These are data that we published in Cancer Cell some time ago. These are studies from an animal model of prostate cancer in which we vaccinated the mice, either with or without cancer. So this is a control group with no vaccine. NT stands for no tumor. So this vaccine, this is a prostate cancer specific vaccine. And when you're a mouse and you have no cancer, it works quite splendidly. On the other hand, in mice with cancer, the vaccine works almost not at all. These data really are not different than no vaccine at all. On the other hand, if you take animals and you give them hormonal therapy, that is, you castrate them before giving the vaccine, it works quite splendidly, as if the mouse had absolutely no cancer. So the idea is maybe vaccines will work better in prostate cancer if we use them early on in the setting of hormonal therapy to maybe get rid of the tolerogenic antigen, or maybe to release the antigen in a way that activates those antigen-presenting cells that we were talking about in the beginning. This is being tested in a trial that's close to accrual now using the vaccine Sipuleucel-T. In this trial, men with biochemically recurrent prostate cancer, not metastatic, not treated with hormones, are randomized to receive either the vaccine followed by the hormonal therapy or the hormonal therapy followed by the vaccine. They got the hormonal therapy intermittently for only a year, and we're now following the immune response. We're actually looking at the patients, a percentage of patients in each arm that have recovered their testosterone but remain PSA progression free. And these are data that we hope to accrue by early next year. So, in summary, and I think I'm going pretty close to on time. You know, the world of prostate cancer is getting a little bit more confusing. Currently, first-line therapy is usually hormonal therapy, seen on the left. Sipuleucel-T is approved for men with metastatic castrate-resistant disease. Usually docetaxel is given after that, but it's possible to do other sequence. As you're well aware, new hormonal agents have been approved, both in the pre-chemotherapy, like over here, and in the post-chemotherapy setting. These are drugs like abiraterone or enzalutamide. These are excellent drugs. They have a high response rate. They produce a survival benefit. Ipilimumab is not going to be approved here in the post-chemotherapy setting. I showed you those negative data. We're waiting to see if it will be approved potentially in the pre-chemotherapy. Same setting, in fact, as ProstVac VF. Other agents are approved, including the alpha emitter alpha-radin and another taxane-based chemotherapy, cabazitaxel. So the bottom line is, if you look at this whole picture, it's a little bit complicated. And we're not really sure. There are not really a lot of large randomized phase 3 data sets. In fact, there are none that can really point to us the best overall order. But I can tell you from studies in animals and from general immunological principles, it generally winds up that anti-tumor immunotherapy looks like it works better earlier on in the disease. And so in summary, cancer vaccines, these are ideas of pressing on the accelerator. One is FDA approved in prostate cancer, and one, ProstVac VF, is in a large randomized international phase 3 trial, and we're waiting for the results of that. In terms of blocking immune checkpoints, I should mention another immune checkpoint called anti-PD-1 was tested in a small cohort, about 17 patients of men with metastatic castrate-resistant disease, and objective responses were not seen. That doesn't mean that blocking PD-1 might not work earlier in the disease state. It's something we're trying to initiate in my lab and with other groups. Taking off the brakes by blocking CTLA-4 has a little bit of evidence for activity provided by my friend Dr. Slovin. And again, a phase 3 randomized trial in the pre-chemotherapy setting is currently ongoing. The one ace in the hole we have for prostate cancer is that androgen ablation is immunologically likely our friend, and it's likely that combination approaches using immunotherapy, even second-line immunotherapies like enzalutamide or abiraterone, could add to the hormonal therapy, to the immunotherapy that we're discussing. But with that, I'd like to finish my part, and I think the idea is to open the floor for questions, which you're theoretically supposed to type in, and then the moderators will present to me.
Great. Thanks so much, Dr. Drake. That was very enlightening. It's good to see that there's quite a bit of activity going on in the pursuit of immunotherapies for prostate cancer, and I'm glad to see that we're getting different options. So we do have a few questions. I'll begin to pose these to you. These are questions submitted by others in the room. If you have a question for Dr. Drake, now's the time. You can type it into the Q&A box on your screen, and we'll try to get to as many questions as possible. So the first question, Dr. Drake, is what is the relationship between PAP and cancer? Is it found only on cancer cells, or is it also found on normal cells?
It's a great question, actually, and to be at the risk of being a little bit flip. In one sense, it doesn't matter all that much in this setting because in the clinical trials that have been performed, all these men have had primary treatment. So they've had either radiation therapy or they've had surgery. So the only PAP in these men in their bodies is in cancer, actually. But the question is, frankly, a very good one. So PAP is expressed in normal prostate cells as well. So if that vaccine were to be given early in the treatment setting, say before surgery or radiation therapy, then there's a possibility that it would target normal prostate epithelial cells. The prostate cancer, though, honestly, we have actually one advantage that maybe other cancers don't have, and that's the prostate cancer. The prostate gland is not required for normal life, frankly. So if you had an autoimmune reaction and totally destroyed the prostate, you would still be alive. This is not the case for something like the lung. So off-tumor effects in the prostate are probably a little bit less worrisome in vaccine approaches than they are in some other cancers that involve vital organs.
Okay. Is there a reason to think that ipilimumab would be less effective for prostate cancer than for melanoma based on the types of cancer antigens present in the two tumor types?
It's interesting. So there is reason to think that prostate cancer is a slightly more challenging immunological target than other cancers. And there's a couple reasons. Melanoma, in sharp contrast, is a pretty good target for immunotherapy because it has a high mutation rate. We think that maybe these mutations give rise to new antigens that the body hasn't seen before and that can be recognized as foreign, actually. So melanoma has that advantage of a high mutation rate. It's much higher than prostate cancer. The other thing, though, frankly, is that the prostate tumors are in themselves a little bit more immunosuppressive in terms of their environment. There are high levels of TGF-beta, which is immunosuppressive, lower levels of T cells than in melanoma. So even despite that antigen part, there are data that the prostate might be a little bit harder to target. And in my lab, we haven't published all these data yet, but if you take T cells out of the prostate and you try to activate them, it's very, very hard. This has been published by my friend Vincenzo Bronte as well. And it turns out that those cells are a little bit more refractory. So I think what we're learning is that prostate cancer is probably a little bit harder than other cancers. Nevertheless, ipilimumab or anti-CTLA-4 is a very potent approach. So we're hoping that in phase 3 setting, it will have some activity in prostate cancer. I should also mention that there have been some case reports and objective responses seen to ipilimumab in prostate cancer. So I think there's a good potential for this agent. But again, it's not going to be as easy as well.
Right. Why do hormone-sensitive tissues like the prostate and breast seem to get cancer more often than non-hormone sensitive tissues?
It's a wonderful question. This is a very good basic biology question. And you know, there are some data that this has to do with the fact that these tissues, particularly breast epithelium, in women who are going through cycles of normal endocrine cycles, proliferates and then retracts, proliferates and then retracts. And the idea is those repeated cycles of proliferation give more chance for a genetic mutation to arise that leads to cancer. And that makes a lot of sense for like breast cancer, right? But for prostate cancer, that doesn't make so much sense because men aren't going through those cycles of immunotherapy. So why prostate hormonal tissue is more sensitive to immunotherapy is actually a little bit more mysterious. And you know, this is the subject of some ongoing laboratory studies, but it's a wonderful point. And, you know, it's really not well known exactly why this occurs.
I wonder if this is a related question, but why is there such a high incidence of prostate cancer in men?
Yeah, it's a good question. The incidence, and we didn't mention this at the beginning of the talk, the incidence of men in the United States is 1 in 6 of men in the United States, or about 200,000 men a year are going to be diagnosed with prostate cancer at some point in their lifetime. There are some data that this is influenced a little bit in the United States by things like diet and lifestyle. And it turns out that risks of prostate cancer are much lower, particularly in Asian countries. But when Asian folks emigrate to the United States, within a single generation, they adopt the same risk, nearly the same risk as native folks living in America. So there are certainly some diet and lifestyle risks that are accompanied with that. And again, maybe the gland, just because of its location and exposure, is more susceptible to things like
that. Do you think it's relevant to observe the percentage of antigen-specific T cells in peripheral blood? You mentioned taking T cells out of tumor tissue, but in the blood. Or do you think that all tumor-specific T cells are anyway in the tumor and we can't detect them in peripheral blood?
Well, this is an ongoing debate that we have sometimes even with the reviewers of our papers, right? So if you could imagine that you had a really good high affinity activated anti-tumor immune response, it can cross normal tissue barriers. And if it was really high affinity, what it ought to do is it ought to traffic to the tumor and not be detectable in the peripheral blood. Nevertheless, the field is a little bit driven by the idea of infectious disease vaccines, right? So if you vaccinate against something like Hep C or hepatitis B, then you can detect specific antibodies and T cells in the peripheral blood. I think, quite honestly, that there have been very few studies where the detection of T cells, particularly T cells in the peripheral blood, really is correlated with the efficacy of a particular agent in cancer immunotherapy. So I think it's a great concept, but I think that if you're really looking for the action, I think that the tumor reactive T cells hopefully should be in the tumor. Nevertheless, the peripheral blood is easy, right? We can do leukapheresis, we can gather vast quantities. In some of our studies at Hopkins, we take 100 mils of peripheral blood, we purify out the lymphocytes, and we try to find those tumor-specific T cells. But again, I just have to say that in general, it's not so clear that the presence of cells in the peripheral blood is going to correlate with the outcome of the vaccine.
Okay. Let's see, we're getting a lot of questions. These are great. Are there any studies indicating that the use of these vaccines pre or post-surgery to prevent relapse? So are these vaccines being used pre or post-surgery to prevent relapse?
Whoever asked that question, I could say, thank you for asking. So we have an ongoing trial. So quite frankly, I think that's a wonderful idea. And you have to remember, surgery is very effective, right? So basically, you take out the gland, which has all the antigens and all the cancer if it's a localized disease. So this is a great way to get rid of tolerance, right? So we have an ongoing trial right now where we take men before surgery. These are men with high-risk disease, and they're treated with a vaccine. This is a different vaccine, which I didn't have time to go over here. It's called GVAX. It's based, coincidentally, on GM-CSF. So men get GM-CSF-based vaccine, and they get hormonal therapy, then they get surgery. So this is a vaccine designed for two things. One, clinically, is we hope that these men have less of a chance of relapsing. That's really the goal, actually. But the other thing that's great is we have, and as a scientist, this is great for me, is we now have their gland and their tumor in hand, right? And we can see if this approach led to T cells, particularly specific T cells, infiltrating the gland and maybe destroying some of the tumor cells. The other thing we can do with these samples is try to figure out which immune checkpoints are important in prostate cancer, which is very hard to do otherwise. So this is a great trial. It's ongoing. It's randomized. Men either get the hormonal therapy alone or the vaccine plus the hormonal therapy. It's a small trial, though. It's about 30 patients. We expect to finish by the end of the year.
Aside from checkpoint blockade, what other strategies are currently being explored to increase the immunogenicity of tumor cells at the prostate cancer site in the midst of such an immunosuppressive environment?
Yeah, one idea that's really moving forward in melanoma in particular, and I think we're starting to see a little bit of this in prostate cancer as well, is the T cells in the tumor, as someone asked before, are more likely to be tumor specific, right? So if you could take the tumor microenvironment and turn it into a pro-inflammatory space, then you might be able to activate some of those cells right there in situ. So the idea is if you inject an inflammatory substance either into a melanoma or into the prostate gland in the presence of cancer, will that work? There's an ongoing trial with an agent called Prostate, a sexy name, right? So this is injected during the time of radiation therapy to try to change that hostile microenvironment to one that's favorable for T cell activation. And there's lots of different substances that people are checking in that context. There's one in melanoma called TVAC. People are using substances like bacterial products, viral products. So I think that's an approach that's coming forward, frankly, in melanoma, but not quite as much in prostate cancer as we'd like to see it. But it's coming.
Okay. We have time just for a couple more questions, and then we'll wrap up. Is ProstVac considered an oncolytic virus vaccine? Does it work by selectively infecting cancer cells and causing them to die?
Yeah, that's a perceptive question, and the short answer is no. So ProstVac actually is more of a standard vaccine that works by delivering its antigen to those antigen-presenting cells, which then activate the adaptive immune response, CD4 cells, CD8 cells, mostly in that respect. It's not the kind of vaccine that homes to the tumor cells, infects them, and then makes them undergo lysis like these oncolytic viruses, Coxsackie virus and adenoviruses. Some of those do that. But that's not what ProstVac is. ProstVac is more like a standard vaccination.
Okay. Did any of the monoclonal antibody studies first expand tumor-specific T cells before testing anti-CTLA-4 or PD-1?
No, it's a wonderful idea, actually. And the idea there is particularly in hematologic malignancies, there's a great deal of work that's going on in which T cells are the T cells, not the antigen-presenting cells. The T cells are expanded outside the body or cleverly engineered and then reinfused. In some of those cases, when the T cells come back into the body, they turn off again by expressing immune checkpoints. So a good idea is to combine T cell therapy along with immune checkpoint blockade. This turns out to work quite splendidly well in animal models. We're actually doing some of these studies now. But to be fair, it's not been tried in prostate cancer. My friend, again, I mentioned her several times, Sue Slovin, is leading a trial of T cells in prostate cancer. They're targeted against PSMA, but it's early days, actually. They've only treated a handful of patients. So the idea of combining this with checkpoint blockade, which is a very good one, whoever said this was a very good one, is still something that's going to be for the future.
Okay, and just a last question. We do have an international audience, and some are asking when or if Sipuleucel-T will become available in other parts of the world, such as Europe or Asia. Do you know that?
This is a complicated process, actually, and from what I understand, there was the ability was the idea of setting up some manufacturing sites and having it be available, but there were still some issues with the national health programs actually paying for it. And so I'm not really aware of the current status of that, but I'm pretty sure that there was approval for it to be given and perhaps marketed, but not, I'm not sure what the reimbursement policy is.
Right, very good. All right, well, that's all the time we have for questions. Dr. Drake, thank you so much. Once again, I'd like to thank our sponsor, Dendreon, for helping us support this webinar series. Next month, we will have a webinar devoted to breast cancer, October being Breast Cancer Awareness Month. Breast cancer, the latest target for immunotherapy with Lecia Emmons, also of Johns Hopkins University School of Medicine. So you can register for that at cancerresearch.org/webinars. Also, if you happen to be in the New York City area next week, we'll be holding our 22nd Annual International Cancer Immunotherapy Symposium with a CIC satellite session focused on surrogate endpoints for cancer immunotherapy trials. You can register for the symposium at cancerresearch.org. Also, for those of you interested in getting more of a lay landscape on what's available to patients and caregivers on the internet, you can come to our new website, theanswertocancer.org. It's a website devoted especially for patients and caregivers. Also, I'd like to point out that later next month, we will be launching a section on lung cancer. So if you're interested in lung cancer immunotherapy, please be sure to come check out the website then. Finally, next June, for our 3rd annual Cancer Immunotherapy Month, we will be starting our first inaugural Ride to Conquer Cancer here in New York. It's a 2-day, 150-mile epic bike ride. You can register for this at ridetovictory.org/New York. All proceeds from the event benefit Cancer Research Institute. And with that, I'd like to conclude today's webinar. Once again, thank you, Dr. Drake. Thank you all for participating, and we hope to see you next month. Goodbye.
Thank you.